get to have this opportunity to be here tonight because of the special collaboration between Town Hall and the UW Engage program. Together, they host a series called UW Science Now as part of the Seattle Science Lectures. Sponsors include Microsoft and KPLU. Each speaker's program will run about 25 minutes, which includes 5 minutes for your questions. We ask that you come to the mics at the edge of the stage so that we can pick you up for the recording that we're making. Before I introduce our first guest, though, I'd like to mention some other events from the speaker series. First of all, please join us tonight for the Institute of Systems Biology panel, who will discuss emerging technologies in health tonight at 7:30 following these Engage talks. Also, the next two talks in the UW Science Now speaker series will be right here next Wednesday, April 13th, starting at 6 p.m. I will talk about climate change, past, present, and future, and Jillian Lyles will discuss reshaping Western Ocean stewardship. And now to our first guest. Around the world, climate change, pests, and droughts are reducing crop yields, threatening food security, and causing more and more people to go hungry. Arjun Khakhar, a UW bioengineering PhD student, thinks that GMOs may provide the solution to these problems. He'll explain why and briefly introduce the new systems he's building to make plants more resilient to these threats and more productive. Please give a warm welcome to Arjun Khakhar.
Okay, thank you so much. Let's see, okay. Thank you so much for being here and for coming out and taking the time to listen to me. As Elisa mentioned, today I want to tell you about why I think we need to design better crops and how I think we can go about doing that. But first, I need to explain how I started thinking about this problem. So I grew up in India, in a city called Bombay, and you can see it circled right there. And India is a beautiful, complex country. It's filled with amazing places and just fantastic culture, cool people. But like every country, it has its dark side. And I was exposed to one of these dark sides every day on my way to school. And this was it. It was hunger in the faces of the urban poor. And it boggled my mind as a child that we could call ourselves a civilized society and yet allow injustice of this magnitude to exist. Why can't we just make more food so that these kids don't need to go hungry? Is something that I used to think. And just the fundamental injustice of this situation really inspired me to try and do something to fix it. Since then, I've gone to college and now I'm in grad school, and I've learned a lot more about the problem of global malnutrition, and I've realized it's a much more complex problem that's solved by much more complex solutions than simply making more food. In the course of my college and graduate career, I've realized that even though the current food allocation status quo is intolerable, it's potentially fixable through strategies like better infrastructure for the distribution of food or less corruption in the regulation of food prices. Unfortunately, in the course of learning this, I learned of an even graver problem that's looming, that has the capacity, that we don't have a good solution for, and that has the capacity to decimate food security worldwide. And that's what I want to talk to you about today. So everyone in this room knows that the world's population is increasing rapidly. That's not news to anyone here. But how many of you knew that we need to meet the needs of this rising population, we need to increase our food production by 40% in the next 35 years? If we don't meet this goal, we face widespread famine. And not only do we need to increase our food production, we need to do so in an environment where traditional approaches to increasing food production, like fertilizers and pesticides, are showing declining efficacy. We need to do so in an environment where pests like insects and weeds are evolving more and more rapidly thanks to the indiscriminate use of pesticides. We need to do so in an environment where the soil is saltier than ever thanks to the indiscriminate use of fertilizers and droughts brought upon by climate change. Essentially, we need to increase our food production in a rapidly changing environment. And make no mistake that while this problem will affect the poorest and most helpless of us first, if we don't do anything about it now, it has the capacity to consume us all. So what we need is not simply more food to help those who don't have it right now, but more food security so that our rapidly changing environment is no longer a threat to us. So how do we design better crops to meet the needs of this rapidly changing environment? Well, one approach is we could consider what our ancestors did. The human population has been steadily increasing for a long time. So our ancestors needed to produce more food to meet their needs as well. How did they do it? Well, they used a process called selective plant breeding, and I'm going to explain how that works with an example. 10,000 years ago in what is now Mexico, there was a weed called teosinte, and you can see it up here on the left. It was an incredibly bushy plant. It had these tiny fruits that you can see compared to a quarter there. They had these rock-hard kernels that they literally needed to use rocks to smash into this coarse, gritty flour. Essentially, it was a bad crop. But 10,000 years of selective plant breeding later, it turned into what is now modern corn. And you can see it's got much larger fruits now with kernels that are much softer. It's a much better crop. So how did this happen? How did this work? Well, to understand that, what you first need to understand is that every living organism is made up of a set of biological parts that are encoded by its DNA. And these parts specify everything about it: how it looks, how it behaves. And for the remainder of this talk, I'm gonna abstract away these biological parts as these sort of colored blobs in a circle. So here, each of these blue blobs represent one of teosinte's biological parts. So returning to our story, these farmers were cultivating teosinte, and what they found was that not all the plants were the same. Some of them were taller, some of them had softer grains or potentially grains that tasted better. And the reason for this is, over time, the mutations accumulated in the DNA of these plants. And because of those mutations, the parts that were encoded by that DNA slowly changed as well. And so you can see here, some of the plants have red parts or green parts now. And what the farmers saw were that some of the plants were better, they had softer grain. And so they saved the seeds from these plants that had the better traits and used them to plant for the next season's harvest. And in this way, they selectively grew plants that had better biological parts without knowing anything about the biological parts themselves. And so 10,000 years of this selective breeding later, what you got was modern corn, which had a new set of biological parts. And this process is known as plant selective breeding. So problem solved, right? We can all go home. This was the shortest talk ever. Unfortunately, no. And for one simple reason, it's just too slow. So as I said earlier, we need to improve our food production by 40% in the next 35 years, not the next 10,000. So traditional approaches will just not do anymore. We can no longer rely on nature to come up with the solutions to our problems for us and then count on stumbling across them. We need a new approach. Luckily, for the past 10,000 years, in addition to doing a lot of plant breeding, we've also spent a lot of time learning about the biological parts that actually are in organisms and make them function the way they do. And this is going to become incredibly important a little bit later on. But I just want to take a second here to say this is why we fund fundamental research and why you should all be all about that. But sort of coming back to a more relevant topic here. So we know a lot about these biological parts. So for example, we know the kinds of parts that allow plants to sense things that happen in the environment, like an insect landing on them, or parts that allow them to respond in very specific ways, like by producing a particular chemical. More recently, we have discovered how to apply engineering principles to biology so that we can actually design new biological parts that behave in the way that we want them to. And then we can directly add these parts into our plants by adding DNA that encodes them into the plant's DNA. This is sort of analogous to the process of natural variation that's used in plant breeding. There, you wait for thousands of years for random changes in the DNA to slowly accumulate to the level where one part changes into another. by designing our own the biological parts that we need, and then directly putting them into the plants, we actually leapfrog this process, and this allows us to do things much faster. And this strategy of designing your own parts and putting them in is called synthetic biology. So I've told you that we know a lot about all of these natural biological parts. I've told you that we can design our own new biological parts that behave in the way that we want them to. But how does any of this let us design better crops? How do we increase food production or improve food security? So to tell you about this, I'm going to tell you about two projects that I'm currently working on at the University of Washington that try to bring together these two ideas to sort of address some of these problems. So I told you earlier that insects are a major problem for agriculture. They account for 15% of, on average, of crop losses globally, and for certain crops like cotton, the losses can be as high as 80%. So they're a major problem. Recently, researchers have an innovation that has dramatically dropped the amount of crops that are lost to insects is the expression of insect-specific poisons in plants. Researchers found that by adding a biological part, which is sort of shown by this blue rectangle here, they could make the plants poisonous to one very specific insect but harmless to everything else. Unfortunately, there is a caveat to using this strategy. As you can imagine, every plant in the field is constantly producing this poison, but not every plant in the field is being constantly attacked by insects. And as a result of that, a lot of resources, the nutrients and energy, are being wasted and are not available for the plant to do what we want it to do, namely produce food. In fact, recent experiments have shown that this strategy has the capacity to decrease the maximum amount of food we could be getting from these plants by up to 25% for certain crops. So at the University of Washington, I'm developing a new biological part, shown here as this pink rectangle, that connects the plant's perception of insects in its environment and attacking it to the production of this plant insect-specific poison. And thus the plant would only produce the poison where and when it's required, thereby saving resources and potentially increasing the amount of food that we could get from these crops. And so now to sort of go into a little bit more detail and tell you exactly how we designed this new biological part, I need to tell you about two ideas. The first is about how plants actually sense insects in their environment. So when an insect attacks a plant by sort of mechanically damaging and chewing on it, the plant produces a chemical called jasmonate isoleucine, which you don't really need to remember. For the rest of the talk, I'm going to represent this chemical as this sort of turquoise hexagon. So in the plant, there are some natural biological parts. That's the red, it's the red blob over here, that this chemical sticks to, and when that happens, these parts get chopped up and degraded. So that's one of the ideas. The other is an idea from synthetic biology, and it's called a synthetic transcription factor. What this is it's a synthetic part that we made that acts sort of like a switch that lets us turn other parts on and off. So for example, let's say you have a blue part here producing this purple molecule. In the presence of this yellow switch, the blue part is turned off and you don't have any purple molecule produced. So what we did is we took these two disparate ideas and put them together to make a new biological part, the red rectangle. And basically what this is it's a switch, but it's a switch that gets chopped up in the presence of that insect-triggered plant molecule. So the way that this system would work is in the absence of an insect that switches around, and so the blue part is turned off. But when an insect lands on the plant and starts attacking it, the plant produces this chemical that then sticks to the switch, causing the switch to get chopped up. And when there's no switch around, the blue part can get turned back on, which allows for the production of that insect-specific poison. The insect eats that, and then it dies. And so in this way, the plant would only produce the poison where and when it's required. And just to remind you, what that would allow is resources to not be wasted and potentially more food to be produced from the same crops. Okay, the second project I want to tell you about has to do with salt in groundwater. So you may remember a while back I told you that the indiscriminate use of fertilizers and droughts brought on by climate change are major problems for agriculture. One of the reasons is that both of these processes have dramatically increased the amount of salt in groundwater. In fact, this is affecting 20% of the world's irrigated land right now, which is an area roughly the size of France, and that number is only increasing. And it's causing $27 billion of agricultural losses every year globally. So it's a huge, huge problem. Recently, researchers have found a new biological part that acts like a salt pump. And you can see it as that blue rectangle there. Basically, what it does is it pushes salt out from inside the root of plants back out into the soil. And they found that when they put this part into plants, the plants actually can grow on soil that has much more salt in it than they ordinarily would be able to. Unfortunately, there's a caveat to using this approach. So as you can imagine, if you have a static amount of pump present in that plant, if you have fluctuations in the amount of salt in the soil, which is almost guaranteed thanks to climate change, then if you have too little salt in the soil, the pump will be pumping out too much salt from the plant, it won't have enough and it'll die. And if you have too much salt in the soil, the pump won't be able to do enough, the plant will have too much salt and it'll die. So currently I'm developing a new biological part, shown here again as that pink rectangle, that connects the plant's perception of salt in the groundwater to the activation of the pump. So that when there is less salt in the soil, there'll be less pumping, and when there's more salt in the soil, there'll be more pumping. And in this way, the plants will be able to adapt to fluctuations in the salt in the groundwater, and this will give us more food security in the face of droughts. So, to summarize, today I've told you about some big, big problems. I've told you about insects, about drought, about climate change, and of course about hunger. But hopefully I've convinced you that these problems are not insurmountable, and that designing better crops using plant synthetic biology is going to play a major role in addressing them, and that it is not the fate of the poor to remain hungry. So with that, I would like to thank my parents, my advisors, my funding sources, Town Hall and Engage for having me, and of course all of you for coming out and listening. And yeah, I'd love to take any questions. Thank you so much.
Hey, I had a question about so there's going to be a shortage of food.
Mm-hmm.
What role does vegetarianism take in? So we spend a lot of food that people could be eating to feed animals. If we literally just had a large segment of the population to change what they ate, we need to put all this food into feeding animals and be used to feeding people when there's this much technology.
I think that's, thank you so much for the question. I think that's a great question. And so I think there's a lot of science out there that says that, if we all shifted to a vegetarian diet, the world would be better for it. And as a plant biologist, obviously, that's great. Definitely eat all the plants. Makes my job more relevant. Personally, I'm not a vegetarian, and I should be. I think the correct answer is yes. I think that the world would be way better off if we were all vegetarian. And I think we can only strive to do that. Yeah. Thank you. Yes.
Everything I've heard about GMOs is always been to take a gene from another organism, like another animal, put it in a plant. Could you say something about, is that what you're doing? How do you actually change the genetics? Do you use genes from another organism, or do you just stay with the plants?
Sure. That's a great question. Thank you. Right. So actually, this gets to something I was hoping someone would ask about, and I'm really glad someone did. This is sort of a hot button issue. Are GMOs safe? And so, just to answer your question first, you're right. So sort of putting it into the structure of biological parts that I told you about. So a lot of GMOs that exist currently in circulation, that's exactly right. They take a particular biological part, necessarily a protein, from another organism, that's generally bacteria, and they put it into plants. And that makes the plant resistant to insects or a particular herbicide. what we're doing is exactly the same thing, but with a very different goal in mind. So we are taking biological parts and we're actually making our own. So we're sort of stitching together many different pieces from different places to get a part that sort of behaves in the way that we want it to, and then putting that into plants. And so it is definitely, you know, something from the outside coming, you know, in. It's not something that the plant has in it already, but that inherently is not dangerous. And so, you know, this is obviously a very big answer that I could give of, are GMOs safe? The fact that I work on them and I'm proposing to, you know, like fight world hunger with them obviously tells you I think that they are safe. But I think what's important really isn't what I think, it's what all of you think after you have, you know, gone through or been informed by, you know, sort of a set of experts. And I don't claim to be, you know, the expert on GMOs or anything, but these 3 publications are fairly recent, and they're really nice because they're not super technical, and what they are is a summary of, you know, the 1,730 different scientific publications that have looked at whether genetically modified organisms are, you know, injurious to human health, to animal health that are fed with GMO grains, to the environment, to you know, wildlife in the environment. And I think that so I've read all of those, and my understanding based on all of that information is that they're completely safe. There is no evidence so far that any GMO that's in circulation currently has, you know, some sort of terrible effect on human health or on animal health. That said, can I imagine a GMO that would be, you know, extremely bad for humanity? Absolutely. And so I absolutely think these things should be regulated. But as such, what is there right now, there doesn't seem to be anything that's bad for you. I don't know, that was a really long answer to your question, I'm so sorry. Yeah.
I was just wondering if once you perfect your technique, your approach for one species of crop and one species of insect, how difficult that would be to translate to different species of crops or other different insects?
That's a great question. Thank you. So for the insects, the mechanism of the plant producing that chemical to in response to mechanical damage, that's mechanical damage. So essentially, it should work for any insect that chews on the you know, the physical plant, like caterpillars. They're called lepidopterans. But there are other insects that, you know, they sort of stick a proboscis into the plant and suck out its sap. It wouldn't work on those. And as far as the plants themselves go, the nice thing about this is that the biological part that does everything, that's sort of a contained unit. So theoretically, it should work in anything you put it into. That may not necessarily be true because a lot of biological contact, sometimes, you know, something from a different plant might talk to it weirdly, but at least theoretically, it should work in any plant. Yeah, thank you. Yes.
So I understand recombinant DNA to be using basically spare parts from one organism, putting it in another. So you're kind of finding off-the-shelf parts, as you call them. When you talk about designing parts, like, where does the sodium pump come from? Is it an organism that already pumps out salt, or can you actually manipulate parts of the genome to get what you want?
Right, that's a great question. Thank you. Basically, what I tried to do is have like a distinction between them. So take this example here. So every time I showed you a blue rectangle, what that was basically meant to signify is that's a part that's from nature. There, what the researchers did was they sort of found a biological part that did what they want. And so, for example, this sodium pump comes from plants that are already adapted to live on very salty water, like marsh plants. All the red parts that I'm talking about there, what we've done is we're actually designing our own parts using synthetic biology. Essentially what that means is we take sort of chunks of other biological parts that do particular things, like, you know, pump or sort of sub-functions within that, and then we sort of join them together in rational ways to get the behavior that we want. So for example, in this red part here, This sort of encapsulates a lot of complexity, but there is a particular system that senses salt in the groundwater in the plant. We sort of join that to another system that allows for this pump to be activated and inactivated in a rational way, so that we can actually connect those two processes together.